Hey guys, क्या हाल चाल नया दिन नई शुरुआत. So as we were continuing the series for rapid revision notes and आज का टॉपिक है माइक्रोबियोलॉजी. So माइक्रोबियोलॉजी से अलग अलग डोमेन से प्रश्न पूछे जाते हैं. The first is sterilization. अब sterilization में there is physical and chemical methods of sterilization. Chemical में there is formaldehyde, gases and liquids. And gases में we have formaldehyde, glutaraldehyde, beta propiolactone, ये सारे. And then we have in liquids chloroxylenol and all, है ना. जो Savlon, Dettol वगैरह में use करते हैं, phenol, it is used as a disinfectant as well, right? So we have different kinds of sterilization methods and they have different principles. Like dry heat is based upon oxidation, moist heat is based upon denaturation and coagulation, radiation is based upon alkylation, है ना. So that means every type of sterilization is made with different kinds of methods and different kinds of principles. So you need to know about the different types of sterilization: dry heat, moist heat, radiation, the temperature at which it is carried out. Like dry heat is carried out at 150 degrees Celsius for 2 hours. Autoclave is carried out at 121 degrees Celsius for 15 minutes for 15 psi. So steam under pressure is another name. Then you need to know about the indicators. So there are different physical, chemical and biological indicators like Brown's tube, witness tube, Bovidic test, Roy's sachet, plastic dosimeter, and bubble point filter test. So for filtration, then you have different chemical methods or biological indicators that include Bacillus stereothermophilus for moist heat, Bacillus subtilis varnigar, Pseudomonas diminuta, Serratia. These are different bacteria that are used for different kinds of biological indicators for sterilization. Then you need to know about pasteurization, which is carried out of two types: high temperature long term, high temperature short term (HTST), and low temperature long term. The low temperature long term is also called as Holder method, while high temperature short term is called as flash method. So it is carried out very fast at 62. Flash is carried out at 72 degrees Celsius for 15 seconds, while Holder is carried out at 62.8 degrees Celsius for 30 minutes. Then you need to know about tyndallization, which is also called as fractional sterilization or intermittent method of sterilization. It is carried out for 100 degrees Celsius for 3 successive days for 20 minutes. So sterilization has different methods that you need to know, right? So you need to know radiation में 253.7 में nanometers per year. We have ionizing, non-ionizing, and non-ionizing we have UV, IR. In ionizing we have CGX, cosmic gamma X-rays. Then talking about the structure of bacteria, virus, you need to know about the flagella. You need to know about role of fimbriae, pili, cell membrane, cell wall, right? All those things, peptidoglycan, teichoic acids, difference between gram positive, gram negative. Then viruses में what is attachment at lactic cycle, lysogenic cycle, difference between them. DNA viruses का example RNA like picorna, toga, Rio, retro, paramyxo, orthomyxo, papo, herpes. Lots of viruses are there. Polio, है ना? So there are you need to know about which virus belongs where. Retrovirus, so Rio virus, है ना? Then staining mid we have gram staining, acid fast, and also you need to know about the primary stain, mordant, secondary stain, decolorizer, like acid alcohol is in acid fast staining. In this you have ethanol, and primary stain is crystal violet and secondary is carbon fission. But in acid fast it is safranin as a primary stain and secondary stain as methylene blue malachite green. So you need to know about CVI complex being retained, which color is bacteria gives what. Suppose gram positive gives purple color at the end, so it is because the complex is retained, right? And in acid fast also it gives red color because of mycolic acid. So acid fast is used for detection of for mycobacteria species. And then you have IMVIC test, indole, methyl red, Voges, proscover, and citrate. So Cosus citrate is used here. Acetyl methyl carbonyl is formed in Voges test. Indole may also be used a particular reagent. So you need to know about all those things that IMVIC test is used for enterobacter species like Shigella, E. coli, Staphylococcus. You can Salmonella typhi. You can estimate those kind of bacteria. So staining is based upon all of these techniques only. Then you need to the microbiological source like Streptomyces venezuelae for chloramphenicol, Orchidaceae for cycloserine, Rimosus for oxytetracycline, Orofaciens for chlortetracycline, and Streptomyces. Lots of Tenebruces for the Canamyceticus for kanamycin, Streptomyces griseus for streptomycin. So there are so many different Streptomyces. And back then you have Micromonospora purpurea, Micromonospora inionensis for cystomycin, gentamicin, and all. And for assays like E. coli is used in the assay of chloramphenicol. Bordetella bronchiseptica is used in the assay of polymyxin B. So you need to know all of these as well. Which bacteria? Bacillus subtilis is used in some assays. Staphylococcus epidermidis is used in neomycin, novobiocin KS में, gentamicin. And so which kind of bacteria? Micrococcus luteus is used in which? bleomycin mate is used. So you need to know about all of those bacteria as well. So microbiology is basically you need to know about the sterilization techniques. It is a prime most value. Then you can also learn about the D value, Z value, and F value. You need to know only keywords. F is unit of lethality or non-sterility. Z value is related to time, is related to temperature, while D value is related to time. Z value is the tenfold change required in the D value. So whenever you hear the word time, you hear the word D value, and whenever you hear the word temperature, you hear the word Z value. So please make sure that you know about these keywords because they are very frequently asked. Trust me, they do ask questions on these values. So don't miss out these values. Right, so that is very important part of the microbiology. Rest is not that important. Like you can study about the flagella staining, Indian ink staining, Fontana staining, and all. Rest is microscopy. So in microscopy, you need to know about the phase contrast microscopy, dark field microscopy, TEM-SEM, the applications. Then definition and this thing, formula for resolving power, magnification power, what is limit of resolution, what is cedar wood oil, what is 100 XP oil immersion lens, all those things you need to know. So make sure that you're well versed with all of this. So that's it for today's video. I hope this video was insightful, and do like, share, subscribe my channel. More such upcoming videos coming out soon. And to get the latest pharma updates, don't miss out the channel. Press the bell icon as well for getting acquainted with a lot of new stuff and latest updates. Thank you so much, everybody.